So please move to the middle as you can. Thanks for making the break quick and for coming back. So it's my pleasure to introduce or to present to you my mentor and friend Larry Corey, who will introduce our keynote speaker.
All right, so I get to introduce the guy whose picture is all over the place. Anyway, David Baltimore is a man who needs no introduction, but actually he's a man who deserves an introduction. So I think because all of us who work in science essentially owe thanks to David. For me, I've had the gift of his friendship and his advice and his wisdom for many, many years. He's been a scientific colleague. He's a colleague in starting Immune Design, a company down the street. And I've been the recipient of his largesse. David trained and helped us recruit Jesse Bloom, one of our faculty members who we are enjoying. But most important, David has been the ultimate example of the need for scientists to be involved in national policies, to shape them, to get the scientific community involved in shaping our national ethical and both financial priorities. So while he's clearly a famous man, and I could actually end here, I'm going to tell you a few things about David Baltimore. So a few facts. Number one, he got his BA at Swarthmore in chemistry. He got his PhD at Rockefeller University. And he got a Nobel Prize 11 years post-PhD for discovery of how retroviruses replicate. He's been president of both Rockefeller University and the California Institute of Technology, and he's currently the president emeritus and Robert Andrews Millikan Professor of Biology. He lives in Pasadena, California, and he fishes in Hamilton, Montana. To say David is an influential scientist is, to me, one of the English language's greatest understatements. One day, when I was feeling good about myself, I looked up my H factor, and then as a barometer of mine, I looked up his. And he made me look, excuse the expression, like a scientist from Dubuque. Now, David's present research focuses on the control of inflammatory and immune responses, as well as the use of gene therapy methods to treat HIV and cancer. He heads a program called Engineering Immunity, which is a cool name. And in addition, he co-directs the Joint Center for Translational Medicine between Caltech and UCLA. But what we need to extol David about is really his influence on scientific policy, and especially his influence in the field of HIV. He is a marvelous spokesman for whatever the issue is, whether it's recombinant DNA or biological warfare, the ethics of genetic engineering. David's advice is sought. But for HIV, we need to remember he led the initial committee on the NIH funding in the response to HIV. He was a major figure in creating the NIH Vaccine Research Center and getting Gary Nabel, one of his eminent trainees, to head the VRC. He helped in the recruitment of the people there, and he was the leader of the Scientific Advisory Board for the first 10 years, and I got to see him exert an influence there. He's also exhibited his influence on the HIV field through the head of an advisory committee to NIH on that, which, even though he's left that committee still 5 years after, is still called the Baltimore Committee. He hasn't been on it for 5 years. So we all listen to when David speaks. So enough. He's a wonderful man. He has a great wine collection. He knows how to fly fish. He has a lovely wife. What else to say?
And I have friends like Larry Corey, which is wonderful. I have a friend here who's going to plug me in, so I can get rid of that. Here's a laser pointer. All right. Okay, I'm now oriented. Thank you so much, Larry. That was an overly kind introduction. And it's a pleasure to be here. It's a pleasure to participate in this meeting. I've already learned a lot and hope to learn more. My own interest, as Larry said, has been through various strategies to engineer immunity. to HIV, to cancer. And those strategies all use some form of gene transfer, gene therapy, and they run the gamut from using gene therapy for T-cell receptors, for antibody genes, for siRNAs, for antigens, in each case target to a different cell population, B cells, T cells, dendritic cells, and by different strategies. And it's all been an attempt to see how far you can go with gene transfer in clinical medicine. A goal that I sort of devoted myself to when I left the presidency of Caltech and was wondering what you do in retirement. And the first thing is you don't retire. The one of these various approaches that I think has the opportunity to be the most dramatic is vectored immunoprophylaxis, VIP. And I think it is a way that we could actually deal with transmission of HIV. And I'll tell you about it. And I should say, and I'll go through the outline in a moment here, that we are awaiting the initiation of clinical trials in this area, whereas in many of the other things that I alluded to, we have clinical trials underway. It has taken a long time to get clinical trials going, and I apologize for those of you who have heard me talk about this before, that I don't have a whole lot of progress to report. So VIP is a way of preventing transmission of HIV, and I'll talk about it and how we validated it in humanized mice with a human immune system. And I'll talk about the extension of it to other infectious diseases because it's fundamentally a platform technology, and consider the real need for reversing VIP as a safety precaution. But of course, this is a symposium about cures. So let me comment for a minute on that context. And so I started thinking when I was putting this slide together about other viral diseases, not HIV. And they really fall into interesting categories. Most viral diseases have a binary outcome. Either the virus kills the infected person or animal, or the person or animal fights it off and is cured by their own immune system. Now, they may be scarred for life by it. Polio is a very good example of people who did cure themselves, but were affected in ways that affected the rest of their lives. And so why is that? Well, actually, it's a little hard to know why you get that binary outcome. But it looks like either the immune system of people who are killed by a virus is too weak, or really more likely too slow to respond. But there are a few viruses, like HIV, whose binary outcome is either to kill a patient or to lead to chronic disease. And as we know, even antiviral drugs don't provide a cure. Others are like herpes viruses, which generally lead to a chronic outcome, polyoma-related viruses, which almost always lead to a chronic outcome, even though we don't know it because they don't have any acute effect. And those are cases where the virus finds an immune privilege site, often the brain, and hides out there for our lifetime. it is the immune system that keeps those viruses in the immune-privileged sites, because if they get out of those sites, they presumably get neutralized very quickly, or the infected cells get killed. And so HIV actually becomes almost unique in the fact that the immune system can't hold it back. And although we're interested in the reservoir, which is the bottom 1%, 0.1%, whatever, of infections, the immune system generally fails on its own to control that virus, and does that for the lifetime of the patient, and therefore it becomes a lethal disease. So what would a cure be? A cure would be, it seems to me, no detectable virus and no probability of virus returning. And I don't think that we're likely to see that for some time. And I think some of the discussions that we heard even earlier this morning show the distance we have to go in order to get to that outcome. But if we could treat patients so that they don't have to take medications, so they're free from the danger of HIV-induced disease, and so they have a minimal possibility, at least, of transmission, I would consider that a win, even if it wasn't a cure. And I think what I'm talking about ultimately could do that. Although I'm going to talk about it in a prevention mode, there are many people working on it, and I'll make that point later, in a therapeutic mode. Now, why is HIV such a unique virus? And it is because it eludes control by antibody, whereas almost all other viruses are open to some control by antibody. And we all know the reasons for that: carbohydrate covering, open surfaces, the protection of the CD4 binding site, the two-step mechanism of entry of the virus, and the covering of the second step site until the virus has gone through the first step. And so there have been very few antiviral antibodies able to neutralize the virus, and fewer still of those that are broad and potent. But the truth of the matter is, all we need is a couple if we use those antibodies in a smart way. So we've had the challenge in front of us since the day that the discovery of HIV was announced, as I pointed out last night to a group. We've had the challenge of making a vaccine, and that challenge remains in front of us. I've been saying since 1986, when I ran the study that Larry was talking about, that a vaccine is at least 10 years off, and I keep saying it. And there are lots of other methods of prophylaxis other than a vaccine that can be thought about. Many of them have been tried, and they've run afoul of patient compliance and the inability to have something on board that the patient, or not patient is the wrong word, that the person will continually take to provide prophylactic effectiveness. T-cell vaccines have so far failed, but, and Dan Baruch will certainly talk about that, there are leads now to the creation of such a vaccine, but we don't know how far that can be carried. And antibodies, I think, are at least short-term the place to start thinking about. And just in the last, 5 years maybe less, Gary Nabel, Dennis Burton, Michelle Nussensweig, others have found that some people make some antibodies that are broadly neutralizing, highly potent, very effective, can be captured as monoclonal antibodies, and can be studied, have been studied in great detail. They don't seem to be much good for the patient because in a patient, there's a continual battle between two evolving systems, Darwinian evolving systems. One is the virus evolving to escape antibody, the other is the immune system evolving to capture the virus. And in that battle, the virus seems to win every time. And so although these antibodies are terrific, they're not good enough to stay ahead of the very creative ways that the virus can mutate to protect itself against neutralization. But they are creating a revolution, these antibodies, because they say that protection should be possible. And if can we use the information from these antibodies to make a vaccine? Maybe. But if we remember that the actual number of particles that infects a person is very small, generally 1, the target that we need for prophylaxis, even if it is highly variable, is a very simple target. And so all we need to do is to get on board a, I think, relatively low level of these highly effective antibodies and prevent transmission of virus. Now, this is HIV. It has on its surface a small number of spikes, each one apparently acting independently because they're so far from one another. And we now know the structure of those spikes in great detail. Actually, we can just I think we can get rid of that. We know the structure of the spike, and we can identify on it the places that antibody binds. And each of these colored areas represents a different site for binding. Carbohydrates are sites for binding. CD4 binding sites are a site. The apex up here is a very good site for binding. And thanks to the work of the people I described, we have antibodies that bind up here and down here and all around. I will only talk about antibodies that bind to the CD4 binding site. And that's not because I believe they're the last word, but because we started working with those and one's enough for the moment. So how can we use these antibodies for prophylaxis? Well, what people are thinking about is trying to elicit them. They're human antibodies. They come from the human immune system. There should be a way of getting the human immune system to make them in each and every one of us, because we all have, to a first approximation, the same genes that are controlling our antibodies. So we need to elicit them. But the elicitation of a single rare monoclonal antibody is something that's never been achieved, to my knowledge. And what it means is creating a pathway of mutagenesis that the immune system can take to get to this endpoint. And most of these antibodies actually have many, many mutations in them. And so that pathway of mutagenesis is a very long slog. Maybe we can design a series of immunogens that will carry the immune system forward from one step at a time to this great endpoint. I'm a little negative about that possibility, largely because I think it should be possible to do that, but I'm not sure it'll ever be possible to do it reproducibly in a population of people so that they all respond with these rare antibodies. The other thing is we can make them in large amounts, and that is now being done, and inject them. This is expensive. It requires very strict compliance over long periods of time, but it is possible, and we'll certainly learn a lot. We are already learning a lot from trying to do it. But we and also Phil Johnson at University of Pennsylvania have undertaken to use viral vectors to program the immune system to make these antibodies. If you can program, not the immune system, we program the body to make these antibodies. Actually, it's not the immune system. And if we can do that, then we can keep a constant level of antibody over a long period of time. And that's what we're trying to do. We call it vectored immunoprophylaxis, or VIP. It's a way of directing the body to make individual monoclonal antibodies, or more than one, because you can inject people in multiple sites, you can make multiple vectors. That's pretty straightforward. And so we're using as a deliverable genes that encode antibodies, not literally a vaccine in which the deliverable is an antigen that elicits an antibody. And we've been thinking about this actually for now quite a long time. The first 5 years of this work was supported by the Gates Foundation. Without their help, we never would have been able to do it. And the guy who deserves the bulk of the credit for this is Alex Ballas, a postdoctoral fellow working in my laboratory, who introduced us to and developed in our lab adeno-associated virus as a vector to do this work. Alex is now at the Reagan Institute in Cambridge, Massachusetts. AAV has lots of very positive characteristics. It's a tiny virus, 20 nanometers across, but it's non-pathogenic in humans as far as we know. And as a vector, if you take the guts of it out and put in the genes you care about, it's non-integrating. So it's DNA, but the DNA actually in an infected cell sits there as a plasmid and churns out product RNA, dependent on what promoter you put in the vector. It makes a lot of product and does it for a long time, because if it infects a cell that doesn't divide, it stays in that cell, as far as we know, for the lifetime of the cell. And cells like muscle cells, brain cells, and others stay in our bodies for very long periods of time without dividing. Virus is easy to grow to high titer by transient transfection. There have to be better methods to make it than that, because that's not a scalable procedure. It has one clear negative characteristic. It's very small, and you cannot put more than about 4.5 kb of encoded information into the virus, into any one particle. The virus itself actually has two inverted repeats at either end of a single strand of DNA that encodes two proteins, the rep and cap proteins. All of that is taken out. All of it is then replaced. Sorry, I don't need that. Replaced by material that we're interested in. And in the vector that we're using now, we have the heavy chain gene, the light chain gene, cDNA, actually heavy chain cDNA and light chain cDNA, linked by a picornavirus 2A sequence with some other things in it. It's actually a virus made out of pieces from 5 other viruses. And we also have a virus with the same promoter. It's a homegrown promoter, the Cas promoter, that drives luciferase. And luciferase is valuable because you can see where it is, and so we can see where the vector is and how long it's maintained in animals. And here we have injected that vector intravenously. If you do that, it goes to the liver. You get a certain amount of production of protein, luciferase protein, over time. But if you inject it into the muscle, the hind muscle of the leg, then you get a much, much higher level of production. The difference between this level of production and that is about 100-fold on this scale. And so we have chosen, and it stays in the muscle very nicely over long periods of time, and so we have chosen to use muscle to inject the virus. I said a long period of time. Here we follow in a mouse for 64 weeks. You can follow it as long as you wish. For the lifetime of the animal, after injection of the luciferase virus, the gastrocnemius muscle produces a roughly constant amount of luciferase. The difference between the peak level and the level 64 weeks later is a factor of 2 or 3. Thus, we did the same thing with the vector that makes antibody, and you again get a peak level a few weeks after injection, and you maintain a level close to that out to 64 weeks, now measuring the amount of antibody in the serum. So the antibody that's made in muscle cells is secreted and is moved and stays in the circulation and at remarkably high levels. So when we optimized this whole procedure and used 4 different strains of mice as recipients, 2 immunodeficient, 2 wild type, we found that they had within ERA the same levels of production of serum antibody, and that amount of serum antibody was anywhere from 100 micrograms per milliliter to 1 milligram per milliliter of antibody for, again, as long as we wish to examine it. That's a very high level of antibody. Let me put it in context. A human or a mouse has about 10 milligrams per mil of antibody continually in their serum. So we're putting in a level which is anywhere from 10% to 1%, the level that's ordinarily there. It's not a level that you would expect to perturb anything, but it is a level that is very rarely, if ever, seen as induced by an immunogen. To see whether that level of antibody can protect against transmission of HIV, and it certainly ought to be able to, we used NSG mice, immunodeficient mice, put into them human peripheral blood mononuclear cells, the very simplest form of providing an immune function to these animals. And basically what we're doing is giving them CD4 cells so that they can be infected. In fact, CD4 cells selectively grow up in these animals, and you get anywhere from 50 to 75, even 90% of the T cells in the periphery as CD4 cells. You wait a couple of weeks for them to grow up, challenge with HIV, sample weekly. If you haven't put any antibody on board, as I've said, you actually get a continual enrichment of CD4 relative to CD8 or relative to the total lymphocyte population. But if you have antibody, if you this is before antibody, just following the cells, the fraction of CD4 cells increases. If you infect those animals, then the fraction of CD4 cells goes down, ultimately to about 0. So if we administer antibody, humanize the mice with engraftment of PBMCs, challenge with HIV, and follow the ratio of CD4 to CD8 by blood sampling over time, what we see in an animal with no antibody is that almost every animal crashes. Occasionally there's an animal that doesn't for presumably technical reasons, although we've tried very hard to avoid that. But if there's antibody on board, and this is an old antibody that has very limited utility, but was the best available when we started this work, the animal, every single animal is protected, and protected for as long as we can follow them, which isn't terribly long because graft versus host disease finally gets these animals. So the question is, how completely protected are these animals? And that's a very hard question to answer. Is this absolute sterilizing immunity? That is this level of antibody so high that even though we've infected these animals with a large dose of virus, certainly thousands of infectious units, that every one was neutralized before it got into a cell and started making product? It's very hard to know that, and we don't know that. But what we do know is that if you search through for any nidus of infection in tissue using an anti-P24 antibody, you can find absolutely no nidus of infection, whereas in animals that are given the luciferase virus, you see the infection very readily. And that's quantitated here as not detectable in B12-protected animals and with a certain fraction of infected cells easily detectable. We also used a number of other monoclonal antibodies in this work, and none of them gave this level of protection. There was basically breakthrough in all of those animals. So that's why we've focused on anti-CD4 binding site antibodies. That's B12 as a CD4 binding site antibody. And we think that gives the best protection. So we have titrated down, and I won't show you a lot of data now, to find out what is the minimal concentration of antibody in the serum that gives protection. And in that work, we found the animals that had 8.3 micrograms per milliliter of VRC01 now, which is a next generation antibody, much broader, much more potent than B12, that it was sufficient to give complete protection against, as I say, a high dose of intravenous virus. But 1.6 micrograms was not sufficient to give protection. It did give partial protection. The curve was shifted to the right, but ultimately all of the animals broke through. So it's somewhere between 1 and 10 micrograms per milliliter is an effective dose in this setting. Now, for humans, the setting is different, the infection is different, comes from a different route, and we don't know, but I think my guess, and actually a lot of people's guess, is that 10 micrograms per milliliter is the dose we should be shooting for. Now, most infections occur at mucosal surfaces. And so to ask whether there's enough antibody getting into mucosal surfaces to prevent infection by a mucosal route, we went to the BLT mouse, which is this more complicated, expensive, difficult model, but in the end has much more infiltration of cells into mucosal areas than the simple PBMC transfer that I described. These are populated with CD4 hematopoietic stem cells, which differentiate in the animal. Dong-Sung Ang, who's here actually, helped us a lot with this. We used in this particular study a different antibody, but it's not one that we're carrying forward because it's slightly artificial, but it's a very effective antibody. And we challenge now intravaginally rather than intravenously or intraperitoneally, which we've done also, using a transmitted founder strain of HIV, Regio C, because we wanted this to be as close to a model of human infection as we could get. So this is a virus that actually was transmitted in the natural course of infection in humans. We use a repetitive low-dose intravenous challenge. It's actually not a particularly low dose, but it's not a very efficient mode of infection. And so the effective dose is pretty low. And we did that 20 times over 20 weeks, trying to infect the animals each time, either animals that are protected with AAV antibody or not. We got the same kind of serum response that we would expect, up to 100 micrograms per milliliter in a few weeks. But we looked now in various places, including a vaginal wash, for antibody, and we were able to detect roughly 1% the level of antibody to 0.1% the level of antibody at mucosal surfaces quite reproducibly. That is, for protection at mucosal surfaces, a lot of antibody. Relative to what's in serum, it's a very little antibody. This is not IgA. It doesn't belong here, you might say. But in fact, it's well known that IgG, which is what all these antibodies are, does get to mucosal surfaces. And we studied this in detail, but one simple piece of data is that if you look with a highly sensitive assay at virus, the protected animals have absolutely no virus in them that we can find, whereas the unprotected animals, most of them became obviously viremic over the period of 20 attempts at intravaginal infection. At least one animal was not infected, even with that time. We originally had used X4 virus. We're now using R5 virus. We know that either one gets protected against. It includes molecular founder strains as well as laboratory strains, and it protects against repeated mucosal challenge. Now, the next thing that I would get asked about before it was done was, how about non-human primates? And the problem is that there is no straightforward, good non-human primate model that models what we're trying to do here with human antibodies, because when you put human antibodies into a monkey, the animal makes a very strong response to the human antibodies. They're foreign. We found that to be true. We couldn't use it. We could make shivs that had the appropriate determinants that would be neutralized. But if you put these vectors into monkeys, very quickly, the monkey makes a response, such a strong response to the human antibody, that the serum levels are minuscule. So this was all work actually done at the VRC by Kevin Saunders, working with John Mascola and Gary Nabel. And what they decided to do was to immunosuppress the animals. And you can do that because we're not actually using the immune system here. We're using muscle to make the antibodies. And so these animals were immunosuppressed for a while. The immunosuppression was taken off just before the AAV was put in. So it's a complicated model. It's been published. You can look at the details. So AAV is put in, SHIV is put in about 5 weeks later. And if the animals have an AAV that's making luciferase, or actually making a control antibody in this case, none of them are protected. They all get infected quite reproducibly. If you put in the antibody, then all but one animal got completely protected. One animal, the animal that had the lowest level, was infected. And we assume that the virus got by the antibody protection. And that animal, obviously, immunosuppression didn't work so well. So we feel confident that this works in non-human primates, although it takes a complicated model to show it. And so we are now transitioning this to clinical development. That's been ongoing for longer than it should have been, but in fact manufacturing is now starting, and we will soon, I hope, have the opportunity to find out what happens in humans. We're using VRC07, which is a sort of second generation VRC01, more potent, a little broader. There are other antibodies that you might prefer to use today. They weren't there when we started this, although we have tested many of the newer antibodies in the humanized mouse model, and they all work to some good extent or other. We've met with the FDA. This is, they will allow this trial to go forward. And the question is, can we get the antibody levels that we've seen in humanized mice, and now much lower but effective antibody levels in NHPs? And is it safe? Now, I said this is a platform technology. There's nothing particular about HIV in this. If you can find a monoclonal antibody that's a way of protecting against any virus infection, you could use a strategy like this. For viruses that are under control by standard immunization, there's no reason to think about it. But actually, there are a fair number of viruses where standard immunization doesn't work. HIV is the most prominent, hepatitis viruses, and others. And actually, influenza virus, because the way we now vaccinate against influenza is about as clumsy as you can get. And other non-viral diseases, malaria, for instance, or tuberculosis, if there were good antibodies, which they don't seem to be. It's a relatively cheap procedure. The virus is pretty stable. It can be used in the developing world, and it certainly can be used for bioterrorist protection. We chose first to go to influenza because just as we were doing this, the antibodies that bind to the stalk of the influenza virus, hemagglutinin, became available. And Jesse Bloom, who's here now, he might even be here somewhere, and Kristen Hung worked with Alex on this. And I won't go into the rationale for it. I think we all know that. The antibodies we chose are antibodies that bind to the stalk here on the hemagglutinin rather than the top of the hemagglutinin, where all natural antibodies go, almost all natural antibodies go, and where mutation is easily accepted, and therefore the virus can mutate against those antibodies year after year after year. The stalk antibodies bind to a region that doesn't vary much and are very hard to mutate around, although I think not impossible. Again, you can get up to the same levels of these antibodies. It takes a little bit of engineering, but not much. And they prevent flu infection in mice. So you measure flu infection by loss of weight. An unprotected animal loses a lot of weight. After a flu infection, a protected animal loses no detectable weight, or maybe a little. There are lethal strains of flu that are protected against. There are weaker strains of flu that are protected against. It is, in fact, a pan serotype anti-flu antibody. So this will work. It has the tremendous advantage, particularly for those of us who are elderly, that it works in immuno-incompetent people, but in particular in older people whose immune systems are maybe not totally incompetent, but not very effective, because it isn't using the immune system. A single immunization, you could have two immunizations that made two antibodies to provide universal coverage and to provide stability. I think it's actually a reasonable thing to think about doing. But What the world needs is not so much an anti-flu antibody as an anti-malaria antibody. And so Kaylin Deal, working with the group described there in Gary Kettner's lab at Hopkins, tried to develop this using two antibodies that were known at the time. They're not the world's best antibodies from various points of view, but they work, and they're known to work to protect against sporozoite infections, the initial infection with malaria. So we made vectors, and again, if you put in different amounts of vector, you get different amounts of antibody going up, in this case, close to 1 milligram per mil. That, in fact, gives you different levels of protection against the lethal effects of malaria in a mouse model at the highest level. Most of the animals are protected, most of the mice are protected. But as I said, this is not the world's best antibody. This is only the beginning. Hepatitis C is another case, and again, these antibodies protect against hepatitis C, as shown by Alexander Ploss working with Charlie Rice some years ago now. And the sort of coolest idea was Steve Brimajohn's. We had nothing to do with this. He got the vectors from us. There is an enzyme that hydrolyzes cocaine that can be brought up to fairly high levels with a vector in the serum of mice. And to quote from his paper, vector-treated mice given 40 milligrams per kilogram of cocaine showed none of the physiological or metabolic fluctuations exhibited by the controls. So it's an interesting form of protection against drugs. Now, all of this is wonderful, but if you have 1 million people that you've protected with a strategy of this sort, you can be almost certain that one of those people is going to have a negative reaction for whatever reason. And we haven't seen any negative reactions. We have no reason to predict them, but life happens. And so we need some way to turn it off. And there's nothing in what we've done that provides that. And remember, I said the vector is very small, so putting suicide genes into the vector is not possible. Putting in a second vector with a suicide gene is not even a terribly good idea in itself, because they generally involve the synthesis of foreign proteins, and so you're going to have to worry about immunologic reactions. And there's reason to think that this will go on producing in humans even for as long as 10 years or more. So we need some kind of reversal. And the one that we chose is to use the Cre-Lox system. So we're putting a piece of DNA, exact double-stranded DNA, into the muscle cells. If you could get Cre into there, then it could recognize lock sites and cut the vector and excise a circle from it and make it ineffective. So we did that. We put lock sites into the vector. We compared it to a control vector with no lock sites. And in a simple test in vitro where we either infected with the AAV or we combined it with an AAV that produces Cre, we got a couple of logs of killing of the vector that has lock sites and very little effect on the vector that has no lock sites. And so the idea works. But this is not the model you want to use. You want to put the antibody producing AAV into a mouse, allow it to establish itself, and then come in with a Cre containing vector in the mouse. And we've done that with luciferase here, and it works pretty nicely. These are animals that get the Cree injection. But most impressively is this experiment where we measure antibody levels in animals. It stays up around 100 micrograms per milliliter in the unperturbed animals. The animals given a Cree injection, it goes down about 4 logs. So you can reverse it. Now, I'm not sure that this is the way to do it, but it certainly provides a proof of principle that it's doable. This is the work of a group of, under Alex's direction, a group of really wonderful technicians who did a large part of the work that I've been describing. Jesse Bloom helped us on the flu work. Lily Yang actually was at that time, she's now at UCLA, was at that time running the whole engineering immunity program in my laboratory. Dan Sangong, I mentioned earlier. We got a great start on all of this working with Jim Wilson at University of Pennsylvania, who showed us one end of AAV from another, how to work with it. And with that, let me thank you for your attention.
No, go ahead.
Do you expect any anti-idiotype response from patients?
I think you'll get a certain amount of anti-idiotype response. The experience with really well-humanized antibodies is that they do give, monoclonal antibodies, they do give a low-level anti-idiotype response but not a high-level response. They don't generally interfere with the therapeutic value of the antibodies.
I wanted to follow up on that in the sense that the antibodies you put in with Saunders et al., which you put in the monkeys, these were fully rhesicized, certainly overconcentrated for rhesicized.
Yes.
And then there was some after rhesicized for variable regions as well. And in parallel, Ron Grosch is publishing real rhesus antibodies going into rhesus macaques. And so another interpretation is that we've got a real adjuvant factor with AAV, with all that DNA and all that protein going in, that's making it worse than passive transfer. Did you go along with that?
No, but but I worry about it. And I think until we've done this with authentic human antibodies in humans, we won't know. But it's certainly the first thing we will worry about. The work that DeRosier did was not with fully simianized, I mean, it wasn't with authentic rhesus antibodies.
They would drive the face.
Yeah. And what I'm hearing is that if you do this with non-human primates with real authentic antibodies, that you don't see that kind of response. But that's sort of not published information.
David, you talked about using this as a platform technology, and that, of course, will require that people get vaccinated multiple times with an AAV vector. Do you have any insights into whether any immune response against your AAV capsid would actually prevent that?
Well, interestingly, the experiment that I showed you with Cree was done with an AAV, a single AAV vector. That we're using an AAV8 with an AAV2 core. We used the same antibody, the same vector to carry Cree, and it worked.
That was in a human, that was in an immune deficient mouse.
That was in a, that's right. That was in a human immune deficient. That's right. No, that's right. We have looked at antibody. I don't think there's a lot of antibody being made. We've looked at it in the, I know where we looked at it. We looked at it in the flu model because there we're not using immunodeficient animals. We're using immunoproficient animals. And we could use a second injection. We could get a boost. So it's not a big effect, but there certainly is an anti-vector. I mean, it's protein. It's foreign. It's going to give some response. Now, there are many, many AAVs. And so were I to be doing this for 2 or 3 different diseases, I would make different different AAVs. And over time, people are learning how to engineer AAV so that you can make artificial AAVs.
So kind of a follow-on question from that. So in the hemophilia gene therapy studies using AAV, they've shown that in NHPs and humans, you don't get the same level of persistence that you see in mice expressing Factor 9 first sort of thing. Are you concerned that you'll be able to, won't be able to get expression of antibodies in humans?
I believe that Catherine High, looking out 10 years in people given, admittedly, vectors that produced very low levels, saw the continual production of marked cells all the way out to 10 years. And that there wasn't a lot of diminution in that.
From the original genesis, there was a decline.
But it plateaued and then continued for a long period of time. So again, I think we've got to just see, and it may be that different serotypes will behave differently. I don't know.
Sorry, Shelley?
I just was going to ask whether you think there will be a need to make different isotypes of the antibody, not just the IgG1.
Well, for the moment, IgG1 is doing terrifically well, and certainly in things like the papillomavirus vaccine, which produces that, it gives sufficient protection vaginally. What? Yeah, we've done work with IgA, and what we observed was phenomenal protection at levels that were so low, it was hard to believe. We've published that. But the problem with IgA is that it's not as stable as IgG, and to get up to these levels of antibody, it's a mixture of the stability of the antibody and the production in the muscle. So it's a balance. And so with IgA, we really don't expect to be able to get up to very high levels. And I'm not sure that we need it for anything. But it's something to go back and think about, because IgA is sort of the natural mucosal antibody.
Another question, maybe.
You pointed out the limited capacity of AAVs to take DNA. Has anybody looked out in the wild or somewhere for larger AAVs, maybe with a larger capsid?
Yeah, I know that Jim Wilson was collecting AAVs from everywhere. He never said he'd seen a larger capsid. Capsids are very hard to fool with because there's such interlocking protein shells that making them larger just doesn't work.
Well, I was thinking of a single mutant, but some related.
Some related virus. I haven't run across one, but it's a very good idea. Since we can get a full antibody in there with all of the accoutrements that we have in that vector, we don't feel an enormous need to make it bigger because you can always make mixed infections with 2 or 3 of them. If you want to use 2 antibodies, the best way I think to do it is to inject them on different sides of the antibody at different places.
But you mentioned the suicide gene, which would have to be in that same.
Right, and if we wanted to put a suicide gene in, then you'd either have to make a mixed infection or find something bigger. But putting in another foreign protein, putting in now a completely foreign protein like herpes virus TK or something, is not a good idea. So I think we're going to try to use strategies like the one I described that don't require putting in a suicide gene. There are some suicide genes that are human. I'm aware of that. And
What about, a lot of people are now saying that with mRNA, delivering mRNA should deliver.
yes, you can deliver mRNA. I'm not sure you can deliver it at the levels that we're talking about to get the kinds of responses you're seeing. But you could do it for Cree, yes. The other thing is adenovirus, because here's a place where you just want a burst of protein and then done. So adenovirus could work. There are other ways of thinking about doing it. Oh, Matt.
So it's really great data on prevention of HIV. Any sense of whether after infection that what levels of antibody might affect the kinetics of rebound, or could this be some sort of adjuvant in the already infected patient?
All right, so can you use this in a therapeutic mode? There are a lot of people, Dan Baruch behind you, and others, now trying to find out what are the opportunities from a therapeutic point of view with these terrific monoclonal antibodies. And what they're finding is that if you make, if you get a number of them on board at the same time, you can actually drive virus down a lot for a long period of time. But the problem is you've got to keep that level up, and of course what you're getting is a cycling of levels. And I think that with AAV, particularly going into a heart protected person, where you can now get the antibody up to a high level, use multiple antibodies, and then release the drugs, that there's a real opportunity there. And in terms of, quote, cure, that's where the opportunity sits.
Okay, I think we should wrap it up. Thank you very much again, Dr.
Yep. Thank you.
So we'll break now for lunch. Lunch will be served right out here when you walk out to the right, and for the students' lunch, it's going to be walk out to the left in the Z conference room. Please be back in an hour.